It's a pleasure to introduce José Leal, who will be giving tonight's talk. Many of you know José well. He is the science director and curator here at the Bailey Matthews. He also was executive director for 17 years and led multitude of important projects, including achieving accreditation for the museum by the American Alliance of Museums in 2010. He's been with the museum since 1996. He received his Ph.D. from the University of Miami, was a postdoctoral fellow at the Smithsonian Institution's National Museum of Natural History. And he served as an assistant editor for Sea Frontiers magazine. He's currently the editor of the Nautilus, which is the leading peer-reviewed journal on malacology in the world. He holds honorary faculty positions at the University of Miami, at Florida Gulf Coast University. He's also a past president of the American Malacological Society and the Conchologists of America. So he knows of what he speaks, José does. And he's also a terrific person to work with and has a strong interest in photography and is a crack photographer himself. And which brings us to tonight's program, which I'm really excited about. It's about two things really. One, and José is of course going to tell you a lot more about it, but something we call the Digital Imaging Project, which is a multi-year effort to create a photo database of the collection, publicly accessible. And then also a special exhibition, a temporary exhibition that opened on Memorial Day weekend, is going to be on view through November 28th, called In Focus: Precision Photography of Extraordinary and Uncommon Shells. And this is show up on our second floor gallery of about 60 photographs, beautiful photographs, as you'll see, of shells from all over the world. And we want to thank a couple of generous supporters of the In Focus project, in particular, the Institute for Museum and Library Services, who are also supporting the Digital Imaging Project, and then Shell Museum volunteers and supporters, Tom and Linda Annesley. Thank you very much. Just a couple of housekeeping things before we get on to the program. Please make sure your microphones are on mute. And if you have questions, this is a webinar format. So if you have questions, please use the chat function, which if you take your mouse and sort of move along the bottom of your screen, you'll see the chat box, and you can type in your questions, and we will keep track of those and get to questions at the end of José's talk. And one last thing I'll mention is that we don't have full details yet, but we are planning a class this fall on photography and photographing shells and other kinds of specimens. So if this talk interests you, if you've got an interest in photography and it's something you'd like to explore more, stay tuned on our website and through our emails for how you can sign up for that class. So with that, I'll stop talking and thank you again for joining us. We'll be back after José's talk for questions, and hand it over to José. Thanks so much.
Thank you so much, Sam. I mean, it's really fantastic to be here speaking about basically two subjects that are really dear to me, evidently shells and mollusks, and secondarily here in this case, photography and photography of objects and shells. So as Sam very well introduced, and I want to again thank him for the kind words, by the way. That's sometimes we forget the things we've done, but good to be reminded. Thank you. It's a combination tonight of showing some of the methodology we use for this project, called the Digital Imaging Project here at the Shell Museum, funded by the Institute of Museum and Library Services, combined with views of beautiful images taken by two top-notch photographers that worked in the project for the past couple of years. and I'll mention them a little later. So first, I'll explain a little bit of the digital imaging project that was funded by the Institute of Museum Library Services to bring about, let's say, at least 4,000 composite images of shells from our collection, about 2,000 species plus, with a focal areas in the Caribbean, Florida, and evidently Sunnyvale and the Gulf of Mexico, including also the Colin Redfern collection. It was donated to the museum in 2014, Bahamian shells. So it's a 2.5-year project that makes it more accessible for remote viewers, looking and measuring and examining shells from our museum collection. And also, as byproducts of that, we are producing a series of items, educational items, like a poster that we're just going to be publishing within the next couple of weeks for sale here on called Cool Florida Shells. We have images now being uploaded to our shell guide, which is part of our Great Museum website, and so on. So let's see how that works. The museum has a collection that now includes about 131,000 lots. And I always like to explain what the word lot means. Sometimes it's a lot of shells, sometimes it's just one, but a lot is a group of samples of the same species collected at the same time in the same place. Let's say you go out and collect 5 Junonias, that's, rainy Saturday here on Sunnyvale. That'll be a lot that you will kindly eventually donate to the museum collection. I'm just kidding, but I want you to absorb the idea, the concept of a collection lot. Here's an image of the collection, and Linda Ansley is standing by there a few years back, and Linda and Tom, as Sam just mentioned, were some of the supporters that helped with the exhibition that resulted from this project. This looks complicated, but I will explain. This is a flow chart of the workflow of the project as presented when we wrote the grant proposal to the Institute of Museum and Library Services in 2018. Maybe I'll go back now. Hopefully you'll have a little time at the end here. I was talking about the workflow for the digital imaging project. The screen that came before was just a screen of showing the collection, and I'll continue from here on. Mentioning the workflow that we adopted in, or that we still adopt in the digital imaging project, going from the left to right. First, we will select a specimen, and that specimen then will be photographed in different views, which we call image acquisition here, just a wordy expression for taking the picture. We will also take images of labels and notes that accompany that specimen in the collection. And last but not least, we keep a log of all the images that we're taking following a series of protocols. They are standard for natural history museum collections. We check the quality of the material. We process that minimally, and it's posted into our catalog, which is a software called Specify that sits in the cloud. Specify is then moved or incorporated into iDigBio, which is a data aggregator that embraces many natural history museum collections all through the world. And here, last but not least, we also have a version of the catalog posted directly in our website, the web portal for iDigBio. And we store all the images in posterity in the cloud as raw and TIFF files. And finally, we, as I mentioned before, we have potential uses as that kind of spawn from that, secondary uses, such as online guides, posters, laminated guides, and all that. So it is a project that it branches in many different ways. This is a page of our catalog showing just the tip of the iceberg, a few images of some of our specimens in the collection, some samples. And if you look carefully, you can see that for each specimen, for each shell photographed, we may display 3, sometimes 4, sometimes 6, 5 views. And this is what you see in our catalog. If you look at our website, and I encourage you to go through the science and research area of the website and click on collection and do searches there. You can do the classic database searches and opt for images, and you may see some of the materials that we have already illustrated in the collection catalog. And this is from our website. I will explain a little bit of the photography as it's been done. We do use relatively sophisticated photographic equipment. We do something focus stacking for micromollusks. Otherwise, a shell that's very small may not have focus throughout that little object. So we do something called focus stacking. This is the group that started and did most of the photos you see today. Patricia Starkey and James Kelly, who are no longer here with us now. We have Chris now, who replaced them and is carrying the project along. The pictures you see today were all done by James and Patricia. This is a view of the photo lab. We have a relatively good computer. We use Capture One software that does live. We photograph directly from the computer. And the camera equipment, we have a standard Nikon D800, relatively old full frame digital single lens reflex, and different kinds of macro lenses for the regular size photography. And then for the sophisticated micromollusk photography, we use an infinity long distance microscope, a very fine optics. It does a lot of magnification and from a long distance. So it keeps a long distance from the object, which is really cool because you can play with lights and you can easily manipulate the object. Don't have to worry about the lens hitting that shell. And finally, we use an automated system for focal stacking called Cognisys Stack Shot. And I'll explain what focal stacking is. When you have an object that is very small that you want to photograph, it's very hard to get focus throughout that object. Here, I found that it would be easier to show this insect instead of a shell. And this is a little wasp. And as you can see, there are 3 images here. And in each one of those images, the area that is in focus is slightly different. Here on the leftmost image, the area that's in focus is on the left. Then in the center here, another area is in focus. And finally, the third one has yet another area in focus. What the system does, with the focus stacking system does, is it finds the parts of the 3 images or 4 or 5 images that are sharp and in focus and uses only that, merges all those good areas into a final result in which the object is all in focus. This is focus stacking. So it's a stack of images that creates a very good final composite image. You cannot tell that this is a mix of many pictures, but that's what it is. So let's start a little here with our little series of images. This is, first of all, I want to explain that you see here the collection number at the museum. The acronym of the museum, it was created when the museum was only Bailey Matthews Shell Museum. The national name wasn't there. So we kept that because that's the way it's been recorded. Here, the technical name of the species and where it comes from. A scale line is graduated in millimeters, and millimeters is 4/100ths of an inch. So basically, this shell here would be probably around a little less than a half an inch. And this is a typical case. I just mentioned focus stacking. The bottom image here is a classical case where we had to do focus stacking. Otherwise, just a little area here being focused. So this is a composite of about 5 images that were stacked to create an image that's totally in focus. The shell itself is a open water, it's called a holoplatonic gastropod, meaning that it spends its entire life drifting in the plankton in open water. And the animal is much larger than the shell. And this is a very, very hard to find kind of little object, but we have a couple of them in the collection here. Continuing, a little land snail that is a paratype, meaning that the author, when he named the species, he used those very specimens, measuring them, and finally deposited them here in the museum collection for posterity. Named by Tom Waters, was a very close friend of the Shell Museum, who named the shell after the sword of King Arthur, Excalibur. Naming shells can be fun sometimes. And this is Tom in the old days when he was working here at the museum for a couple of weeks back in 2008. This is another type, in this case, a freshwater nerite from Indonesia, and named by Tom Eichhorst, who is the editor of American Conchologist, the COA magazine. And Tom deposited all the types, all the main types of his study. He's published in 2 books on the family Neritidae, the nerites. And this is a shell that I think is really cool because it's really dark. The outside is kind of dark, and but the shield there, the opening of the shell, that has the beautiful orange hue. And here are images of the operculum, which is the little trap door that seals the shell shut when the animal moves inside. Well, the next one, some of you know well. This is a one-tooth Simnia. It's actually a shell here from Sunnyvale. And again, a scale in millimeters. As many of you know in the audience, the one-tooth Simnias, they live on top of sea whips, which are relatives of coral. And they can eat the mucus created by the sea whips and the pigment that comes with that mucus. So in this case, we know that the sea whip that onto which this one-tooth Simnia was living was probably reddish or pink, because that's the color of the shell. They tend to absorb the pigments and take the color of that host sea whip. Next, a not so common small little miter from the Keys, from Isla Morada. So this is a very elegant shell. The word pocellum here, Vexillum pocellum, the word pocellum means beautiful in Latin. And that's just what it is, a very special, very colorful, bright little guy. Well, it goes without saying that this Calliostoma here is a fantastic object, a fantastic shell. We have a couple of species here in the Gulf of Mexico. This particular shell came from off Key West a few years back, from 125 fathoms. That's around a little less than 300 meters, or about 900 feet. And it's super elegant. I love the way the colors the lines, the little beads, and it's a super cool object. I have a little lineup of marginalas, and marginalas are really cool. They look like little cowries, but they're different from cowries in that they do not cover the shell when they come out. They are burrowers, they burrow in sand, and the shell is always smooth and shiny. This one here is a white spot marginala, Prunum guttatum, again from the Florida Keys. We have a lot of samples here from the Keys, from Florida in general. The Keys have a huge influence of the Caribbean, more than we do here in the Gulf. And a lot of those shells are super bright, super colorful, and in some cases super glossy and shiny. Another one that became the poster girl for our In Focus exhibit. It's the Carmine marginala, super elegant, typically with about anywhere between 10 and 20 little denticles here, little projections inside the aperture. Species is found in South Florida, the Keys, and the Caribbean. Another marginala, this is the orange marginala, another glossy shell with a very strong hue of orange color. Unfortunately, they are very hard to find in good shape. The pigment here will fade away pretty fast, especially if the shell is on the beach. In one of the few beaches in the Keys, I actually found them. And it lives in shallow water all through East Florida and all the Miami area and down to the Caribbean. This is a label created by Ken and Alice Ph. Super detailed, and Ken and Alice are living here on Sanibel now and come to the museum often. A little oat, as in the oatmeal, OAT, little oat marginala. It's a relatively small. It's the smallest of all the ones I'm showing you today. So it's less than a millimeter. Less than a half an inch in length. And this is one of those species that while the animal is alive, the shell is very, very translucent. In some cases, almost like glass. And as soon as the animal dies, that transparency goes and the shell becomes relatively opaque. And we really don't know why that happens. It's almost like the animal is capable of protecting, putting some sunblock or something that prevents the shell from becoming opaque. This one just from off Key West, Stock Island, in the Lower Keys. I have a trio here, a trilogy of 3 composite images of one of them. The first one of elegant glassy bubble, also from the Keys, from Lower Matacumbe Key. And this is a very special species. I love to talk about it. The fact that they belong to a group of gastropods that all the species are hermaphrodites, so they have both female and male sexual systems, reproductive systems. And the animal also covers the shell completely when the animal is moving and active. This species comes in different colors. The shell comes in different colors, and I love this particular one that has this translucent aqua color. Super elegant. We actually have small posters, 24 by 18 for sale in the store with this species and a few others that I'm showing here today, in case you're interested. This particular shell was collected in 1953, by the way, and as you can see, many of the images that I'm showing you carry the original label, so as to give a sense of the history of that particular sample and show the handwriting of the collector and all the cool stuff that comes with it. Same species now with a different varietal. This one collected by Bruce Scholes, who is a active Sunbelt Shell Club member, and many of you know Bruce, I know. And Bruce likes Bunch Beach, and he collected this in the high tide line on Bunch Beach. Spectacular, kind of a lavender, I guess, color. Very hard to come by that way. So he collected it in June 2019, about 2 years ago, and donated to the museum. Now we have a different species in the same genus, the Antillean glassy bubble, collected in the Gulf at the Bel Air Causeway, I believe in Hillsborough County. And you can see the differences between this particular species and the one that preceded. The elegant glassy bubble has those lines, I mean, like etched lines, spiral lines. This one doesn't. And the Antillean glassy bubble is larger and more obese, you know, it's rounder. Those animals lay their eggs in collar-shaped structures that contain thousands of tiny yellowish green eggs. In our website, in the show guide, we have images of the egg cases of all those species. And now, I mean, a favorite of all times, the common violet snail, the Janthina. This one originating from Don Swenson's collection. Don Swenson just recently donated his entire collection that he made snorkeling and walking the beaches of Palm Beach and Martin County over a period of 25 years. And good records, good historical records of all these species, the largest size species from that area. The common violet snail lives in open water its entire life. So it basically builds a raft of bubbles that they use to move about with the winds. And they feed on special other animals that also live their entire lives in open water. This type of community, or those types of animals, are known as neuston, N-E-U-S-T-O-N. And that means, you know, the larger size invertebrates that live on the surface of the water, farther from the coast in open ocean. In April, March, on the east coast of Florida, you can see them sometimes by the dozens or hundreds that come ashore. That's when they're reproducing. Well then, one of my favorite winter traps of all times. I mean, it's a Gulf of Mexico species, deeper water, the noble winter trap. You know, it's a favorite of shell collectors. It's a beautiful shell, and the proportions are so elegant, and the delicate sculpture of blades that we call costae, C-O-S-T-A-E. Those are typical of that species. It's an uncommon favorite of all times. A hairy triton, it is actually, this shell is not, is missing a little, the tip here, the protoconch is gone, but the photographs are so detailed. I think James Kelly took those images and he assembled them so well together that I had to include. And by the way, all these images are on display in the In Focus exhibit. That was an idea that Sam had just after he came on board in March. He said, hey, let's do an exhibit of some of those. And it really blew my mind when I saw the way they came out. Some of them are, I mean, 4.5 feet wide. So you have to come here and see them if you haven't yet. So the Tritons are a group of very special group of gastropods that live throughout the tropical oceans, and many of those species, such as this hairy Triton here, Monoplex pilearis, they have worldwide distribution. Meaning, if you go to you know, the tropics in the West Pacific or the Indian Ocean, East Africa, South America, Florida, this species is present in all these areas. And one of the reasons why is that they do have very long-lasting open water larvae called planktotrophic larvae. So meaning the larvae that feed on plankton. So, and because they can feed in the larval stage, they live for a long time, sometimes months, and they can cross from one end of the ocean to the other. They can come from West Africa to Brazil and so on. Next, again, I mean, I made the selection of the curator's choices, and this to me is a fantastic set of images of a very common and not so spectacular species, you know, the buttercup lucine from Sunnyvale in West Florida in general. This species has yellow color. In this case, this particular one doesn't have a lot. Some of them will be more yolk yellow, more stronger yellow, but I really like that one because it's so delicate. And James captured the images so well. I mean, captured that delicacy. I mean, the delicate nature of those valves of this common bivalve. So there you go. Interesting species. The green mussel. It's an introduced species that came from New Zealand to the east coast of the US, parts of the Caribbean and the Gulf of Mexico. The species is introduced, but it's not a very aggressive invasive. In other words, it's there, but it's not expanding its range too fast. You cannot see many of them, and it's probably not displacing local species as some of the more aggressive invasive species do. So it's a shell to keep your eyes open for. This one actually is from a little north of here, Englewood. We do have in the collection, we have some from Fort Myers Beach and at least a pair of valves from Captiva. The green collar is typical, very attractive species in what is normally not a very attractive group of bivalves, except for those that like to eat them. I have a couple of scallops. I cannot have any, you know, a talk about nice shells that doesn't cover some scallops. This is the Knobby Scallop. It's a relatively small species from the Caribbean and East Florida. This particular specimen came from off Palm Beach, and it was dredged many years back. Interesting enough, pretty much like the buttercup lucine, the Knobby Scallop also has yellow color inside the valves. So it's always a mystery to me why one would have color in a part of the animal that nobody can see, because it's really inside and it's covered by the mantle here inside. Anyway, I really wanted you guys to see this particular sample. Here we have two very similar species, the ornate scallop and the Santis scallop. And one of the main differences is the type of color. As you can see, this one has a model, you know, high contrast color pattern, and the other one, the Santis, is more monochromic. I mean, it's more like just one shade. There are little differences in the sculpture of the shells and all. Both specimens, and at this point, I'm sure you understand that what you see here are different views of the same pair of valves of this scallop, and likewise here. And I really like the way they came out. You can see the side view of the complete pair of valves and the top view in both cases. Those samples came from Colin Redfern's collection that originated in the Bahamas in Abaco, donated to the museum, and are also included in his magnificent books on Bahamian seashells. Lion's Paw. Had to talk about it, had to show off. This one was trawled many years ago all the way in the Gulf of Mexico off Sanibel. And people can find pieces here on the beach, little fragments, juvenile valves, isolated valves, but never the entire paired, articulated, as we call, joined together pair of valves. A file clam, in this case, the Antillean file clam, which is a super delicate, relatively small file clam. File clams are related to scallops. You can tell that it's kind of almost like a skewed, like a distorted scallops, if you will. And they have somewhat similar life habits. Some of them can swim very feebly, not as well as scallops can. And some of them can form cocoons in which they protect themselves, like pretty much like a little butterfly cocoon. And many of them have eyes like scallops do. A beautiful, beautiful amber pen shell from the Florida Keys. We do not have the amber pen shell in the Gulf here in our beautiful Southwest Florida area. The amber pen shell is found on the East Coast, the Caribbean, and the Keys. And it's a really, really, I mean, it's first time I touched one underwater, a live one underwater. I gently touched the edges here on the top of the shell, and it just bent under my hand. I mean, it's like plastic. Very interesting. When it, evidently the preserved shells, after the animal dies, they lose this flexibility, but while the animal is alive, the shell is kind of flexible. They can bury in the sand down to here in grassy / coral rubble areas in East Florida, the Florida Keys. This one is from Sugarloaf Key, close to Key West. Getting closer to the end here. This is a very common local species, your jingo, a common jingo, Anomia Simplex. And this is one of my favorite species of bivalves for many reasons. It's a shell, a bivalve that has a solid byssus. Many of you know what a byssus is. I mean, we talk about that all the time. Volunteer Joyce Matthews has her beautiful exhibit here in the Museum of the sea silk, which is the byssus of European pen shells. She just gave a program at COA, Colleges of America, a couple of weeks ago about that. And the common jingo has a byssus that's not only, it's solid. This is the top valve here. That's the bottom valve on top. I had it reversed here. And the byssus is attached to the top valve, and it goes through a hole, an orifice on the bottom valve, and finally attaches to the hard structure onto which they live, onto which they attach themselves. So the top valve is easy to find. The bottom valve, not so easy. So most of when people are looking for jingos, they will find the top shell, not the bottom one. You're talking about micromollusks. Here is one, and also it's not only a micromollusk, but is a left-handed micromollusk that you can find if you look carefully. You may find it here in the sand close to the water. It's a Triphora, a modest Triphora, Marginala modesta, and Triphoras live embedded in sponges. And they feed on sponges very carefully. They don't want to kill their host. So the sponge is growing and it eats a little bit, nibbles here and there. You can only find them live if you pry open a sponge, which you probably don't want to do. But this particular sample here, the one interval here being 1 millimeter. So this is probably about 5 millimeters, about 1/5 of an inch in size. And by the way, those images were also done with focal stacking. Another micromollusk, this is a deeper water dove snail called the Diaphanous dove snail. And I love the texture. I love the fact that the shell, it has this sheer, translucent, almost like an Art Deco glass. It's really cool. And a shell, if you look carefully, the image shows something you probably couldn't see with the naked eye. We wouldn't see that with your naked eye, which is this kind of very faint sculpture of which we call reticulated, almost like a little grid that sits on top of the surface of the shell. So this is also from off the Florida Keys. Finally, a couple of micros here, the line here being 1 millimeter, pretty small. So that is 4/100ths of an inch. Very glassy. This particular micromollusk has a shell that really feels like glass. The image is not spectacular. It could be better, but I had to show you because it is, the structure is beautiful. It's like a little piece of glass. And last but not least, I want to show you a shell that is it's an uncoiled sundial related to the beautiful noble sundial. So this is called the Exquisite false dial. And as you can see, the whorls do not touch each other, unlike a normal quote unquote shell. So we call it open coiled. Shell itself is about 1/5 of an inch in size. And I had to, I'm not, I have to let you know that this shell is the favorite shell of our finance director, Ryan Powell. And ever since he told me that, every time I see the shell, I think of Ryan and his exquisite false dial. And again, I want to remind you, Sam Ankerson mentioned the In Focus exhibition. You will see all the images I've shown you today and more. There are about 64 images. The exhibit will end on November 28th this year. So I hope you can make it and visit with the museum, the photography exhibit, Beyond Shells, the living animals, which is getting better every day. I want to show you a couple of pictures, the area, the way we display that. I took the pictures before the public came in, so you don't have a good idea of size, but those are probably 3 feet wide. We have some of them are 4. Here are the marginalas that I've shown you today on the wall that goes to the restrooms, the hall, the back hall of the museum. I will end by mentioning the main funding source for this project, which is the Institute of Museum and Library Services, the collection aggregator that we work with, iDigBio, our Specify software, a group that makes it very easy at a very low cost to us, the state of Florida through grants, Sanibel Captiva Shell Club, also supporting us and the collection efforts of the museum, and National Science Foundation that we have a continuation of this project under the Eastern Seaboard funded by National Science Foundation. So thank you so much for your attention. I don't know if you have questions. I will defer now to Sam. If Sam wants to come back here, I will stop sharing my screen.
Okay, I'm back, I think. Thank you, Jose. And I love it that you mentioned that last slide of the sundial shell. I love it that you mentioned that was our finance director, Ryan Powell's favorite. What we found since opening this exhibit is that, pretty much all staff are moving through there every day, and everybody's favorites, they change from week to week, and everyone's got one. So it's pretty cool. And And I was also struck in, you know, I'm looking at this presentation obviously on my computer screen, which is a fairly small laptop, and obviously you're all looking at it on your screens too. But the focal stacking method that you described, and just the level of detail is just so incredible. Probably even more than you could really pick up if you were, in many cases, if you were holding the shell and looking at it yourself. And so it's great to see it that way. And in the, Jose showed you some pictures, but in the exhibit itself, they're blown up to a size where you can really enjoy all that detail as well. We do have a question. Thank you for the comments on chat. If anyone has any questions, please feel free to click chat and type them in. But there is one here from Gail. And apologies in advance if I pronounce this word incorrectly, not being a conchologist yet or a malacologist myself. But does the glossy look of a shell represent the periosteum? Is that how you pronounce it?
The periosteum.
Aha.
I'm actually writing this here. It's almost there. So no, the answer to that is no. Usually the glossy shells do not have a periosteum. The periosteum is kind of quote unquote bypassed in some of those species. Evidently, many other species of mollusks, bivalves and gastropods will have in some cases, very thick periosteum. The best example is the hairy triton, in which the shell I showed today here previously of that hairy triton does not, the periosteum have been removed by the collector. But in many cases, glossy shells are kept glossy because the animal is able to bury in the sand. A well-known case here in our area is the leathered olive that has the super glossy, it's almost like glass. And it's that way because the animal, buries in fine sand that acts almost as a little abrasive, like toothpaste, to keep that shell clean. I mean, and free of settling parasites and things like that. See, other questions that
I see a question here from Diane, and the question is that poster behind you the one on sale in the gift store? And if Diane is referring to the poster behind me with the shells on it, the answer there is yes. Okay, you are. Thank you. The answer to that is no, not currently, I believe. There is, and maybe Jose can talk about it, there is a new one coming online of sort of a compilation of shells, very much in the style of the images of the In Focus exhibit. Is that about right?
Yeah. Yeah, it's I know you guys will love that because I actually, first of all, I did the posters with a bunch of images from the project and some of the images I had before. And I did that during the few days. I mean, I think the museum was closed by a little longer than a month in 2020, at the infamous lockdown. And I was working from home and I would do regular museum work, catalog and stuff. And then when I was tired, I said, well, I'm going to do that. I'm going to do a poster. And the poster is called Cool Florida Shells. And you have to see it to believe it. It will be, I think Gretchen is, our store manager is ordering that from a local printer for all of you to see.
I have a question actually about micromollusks, and seeing the last few slides of your presentation made me think of it, and also because it's something that people seem to be drawn to when they're in the exhibit, just the idea of micromollusks, very, very small shells. And you referenced some of the specimens that you had images of, but can you talk a little bit about micromollusks and kind of like how small do they get, and are, to what small size are we able to photograph them, and that it's
yeah. Well, that's cool. I mean, I love to talk about them. You have at some point, you have to, put a muzzle on because I really like it. And yeah, it's, well, first of all, Micromolesk, it's kind of a, it's subjective, but there's more or less a consensus that anything below 5 millimeters is a micromollusk. That's the way we treat them in the trade with the collectors, the citizen scientists, in science in general. And that would be 5 millimeters would be about 1/5 of an inch. If you have if you're more comfortable with the imperial system versus metric, it will be more or less 1/5 of an inch. And another thing about micromollusks is that most mollusks are micromollusks in the planet. So if you people who are like conservation biologists, biodiversity enthusiasts and scientists, when they do a survey, let's say in a Caribbean island or a Southwest Pacific island, they will do anything to get all the species that can be found there, and either keep them or not keep them, but take notes, take pictures, and by far, a huge percentage, like 80% more or less, if I can remember correctly, in some cases, will be species that will be smaller than 5 millimeters or 1/5 of an inch. That's where the biodiversity is. That's where the new species that are yet to be found are. We do have them here on Sunnyvale to a great extent. We have some families that are very hard to work with. In some cases, we have some groups that in our collection here, we have for that particular group, 20 plus species that we don't know what they are. And but that's exciting, because if you know what they are, I mean, there's work to be done. There are things to be found, and anyway,
it's great. It's great to have the pictures of them, because otherwise, most of us would really have no other way of knowing what they look like.
Yes.
There's a question from Isabel. Where will the printed photographs go after the exhibit ends? So these are digital images that are here at the museum, and were printed onto a high quality kind of poster paper for the exhibit. So they'll stay here at the museum, but actually, as José mentioned earlier, anybody who would like their own print of any of these images may have one. They're available through the museum store, and basically they can be custom printed and framed and drop shipped from the from the place that does that work. So that's an arrangement that can be made through the museum store. There's a selection of about a half dozen that are framed and at the store as examples, but those aren't the only ones that are available. There's actually any of the ones in the show can be arranged if anyone would like that. Okay, thank you. I don't see any other questions at this time. So thank you all very, very much for joining us for tonight's program. José, thank you very much for a great presentation.
Thanks.
And we hope you all have a wonderful evening, and that you'll come back and join us in July 13th and 27th for the next two programs, and keep in touch. And we hope to also see you at the museum soon. Good night.
Thank you. Thank you.